In this video, we will discuss about glycans and virus, how this glycan helps the virus to penetrate within the cells or penetrate within the host cells. So let us start with the virus structure in general. So this is the virus structure, and it has this cell surface glycoproteins that contains these glycan structures. And these glycan structures has different kind of roles. It can interact with the host cells in two different way. First one is known as the main receptors, where it will favor the viral attachment and the fusion, and the second one is like a co-receptor, or we can say host cell surface lectin, that helps in the that interacts with the glycans on the virus. uh, it interacts with the glycan on the virus and helps in the virus attachment and fusion within the host cells. So this is just a general structure, and this is one of the example that is shown here. In this example, it has been shown the what could be the reason for the glycans to be present at a specific site of the virus. And one of the example is shown here is that the glycans are present at a site where it can shield the vulnerable protein epitopes that can be detected by the host immune system. So this glycan act as a shielding to, that helps in the immune evasion from this vulnerable protein epitopes that can be detected by the host immune system. The next one, the glycans could be present at a site where it helps in neutralizing the glycoepitopes, and so that the glycan adjacent the epitopes adjacent to these glycan sites will not be detected by the will not be efficiently detected by the host immune system. However, where there is no glycan presence, there usually non-vulnerable protein epitopes are found. Those and those epitopes, in most of the cases, similar to the host epitopes. So the host immune system will not be able to distinguish whether these epitopes belongs to their this epitopes is the self epitopes or non-self epitopes. So this could be one of the evolutionary path evolutionary ways by which virus evolved themselves, so that they can prevent themselves from the host immune response. Now let us understand what are the different kind of glycans and how they interact with the host. So one of the type of glycans is known as A1 or minimum glycoepitopes. In this case, glycans directly interact with the ligand. There is no protein presence. However, in the another case, which is known as A2 glycan protein glycoepitopes or GPEs. In this case, both the proteins and the glycan with the protein epitopes and the glycans can interact with the Y part of the antibodies. And the next type of interaction is known as topological glycoepitopes interaction. In this case, the glycans that are present on the glycoproteins and the cluster of glycans then interact with the X shape, then interact with the X type antibodies, X type structure of the antibodies that it can be shown here. So this is known as topological glycoepitopes interaction. In this case, the cluster of glycans, in this case the example shown is about high mannose glycans. It can interact with the X type antibodies. And this part B shows how the normal interactions occurs with the host. So it involves all the three kind of interactions that is shown in the left hand side. And in the part C, this is the example that is shown here about the GP150 glycan interactions with the antibodies. And these antibodies contains is this antibody this structures is usually typically depict the part 3, that is topological glycoepitopes interaction. So this is the GP120 is the glycoprotein that are present on the HIV virus, and these are the glycans that are shown here. And above that glycan, this is the antibodies interaction that occurs with these glycans. So this is how the GP120 can be interacted with the X type with the X type antibodies. And the region shown in the pink color is the variable heavy region, and the region shows in the we can say purple color is the variable light region. So this and this is this antibody is known, this monoclonal antibodies is known as 2G12, is been named as 2G12. So it can detect this GP120 glycoprotein with the help of these glycan interactions, or in other term it is the interaction is known as topological glycoepitopes. Interaction is based on topological glycoepitopes. So this is how the glycans can be used by the virus for fusion and fusion and infection, and also these glycans can be used for these antiviral therapies. And one of the examples is shown here. These antibodies can be used to detect this HIV glycoprotein expressed on the HIV virus through this topological glycoepitopes interaction. However, this area is still on its initial stage, and it needs to be evolved with time, so that this approach can be used commercially to target the virus, so that patients cannot be so that patients can be cannot be infected by those. So the patients affected by this kind of virus can be reduced. So I hope you got an idea how these glycans and virus interactions are important. And if you need to understand more about this glycans and virus, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.